Okay, so today we'll be discussing about microorganisms. Okay, as the word suggests, micro means very, very small. So microorganisms means the organisms which are very small, and these organisms cannot be seen with the naked eye. We need a microscope to see these organisms. Now, in this chapter, we are going to learn what is the role that they play in our lives. What is the role of microorganisms in our lives? Are they useful to us? Are they harmful to us? Or are they useful and harmful both? Okay. Why do you think we need to study this chapter? Can anyone tell me why you think we need to study about microorganisms? Both of y'all, can anyone tell me why we need to study about microorganisms? Why do we need to know about them? They are something which we cannot see. Why do you think we need to know about them then? Any idea?
For diseases,
Yes, Janvi, I couldn't hear you.
purposes,
For which purposes?
medical purposes.
Okay, very good. Okay, for medical purposes, can you something more? Okay, for medical purposes, like because they cause various types of diseases. Okay, and if we and since they cause so many types of diseases, if we don't know the microorganisms that are causing the diseases, we cannot prepare the medicines for them. Okay, for example, now we are all in a lockdown because of COVID-19. If we did not know that what the disease is caused by a virus, could anyone or could Russia have been able to make the vaccine for it? No, it would not have been possible if we would not have known that this particular virus is causing this disease. Okay, so it is very important that we really know about the microorganisms because they cause diseases, and because they cause diseases we can make the medicines for them if we know about them. Otherwise we cannot make medicines, we cannot make vaccines for them. Okay, so that is one importance. Any more importances why we are going to learn this chapter, why we need to learn about them? See, one more other thing is because these microorganisms are also useful to us in many ways. So as to use these microorganisms for different purposes on a commercial level. Okay, like it is used for making alcohol. It is used for making these vaccines. So on a commercial level, to learn about them so that we can use them. It is used for making curd. It is used for making the bakery products. So to learn what roles they play, and to learn what is how they are useful to us, we need to know about them. Okay, and we can use them on a commercial level as well. So this is the reason why we are going to study this chapter. Next slide. Okay, so do you think that we can see everything in this park? Can we see everything in this park?
No.
No, because there are some things that are not visible to our naked eye, which is very less than 0.1 mm. If the size is less than 0.1 mm, we will be unable to see these organisms. Okay, because they're very small. We can see something within 1 mm. Okay, like a grain of sand, a grain of salt, a grain of sugar, a rice grain. We can see these things. It is little bit difficult to see them, but still we can see them. But anything which is less than 0.1 mm, the human eye does not have the capacity to see these organisms. So that is the size of microorganisms, less than 0.1 mm. Okay, so we cannot see them with the naked eye. We need a microscope to see them. All right, next slide. Okay, as we discussed before, that the microorganisms are the organisms that are very small and we can only see them under microscope. These These are the microscopic view. Okay, we cannot see them so big. They are not so big. Only when they are magnified we can see them. Okay, what is the structure can be visible only through a microscope. Next slide. Okay, now where are these microorganisms found? We can say they are found everywhere. They're found in the air, in the soil, in the water. They're found inside our body. They are found in the glaciers. They're found in the high mountain peaks. They're found in the volcanoes. They're found in the hot springs. Okay, they're found in a desert which is very hot. They are found even in space. So there is nowhere that microorganisms are not found. They're found everywhere. Okay, they can sustain anywhere. In space we cannot survive because there is no oxygen, there is no water in space. We cannot survive, but you see these microorganisms can survive everywhere. Okay, there can humans survive in hot springs? No, it's going to be very hot. The temperature is going to be very high. So humans cannot survive in hot springs. But microorganisms have the capacity to survive everywhere. Okay, next slide. Okay, so there are 5 types of microorganisms. The first one is bacteria, algae, fungi, protozoa, and virus. Okay, now bacteria is the smallest microorganism. It is the smallest of all the microorganisms. And bacteria has a single cell. Do you know what are cells? What is a cell? Can anyone tell me? What's a cell? Have you heard about a cell before? Yes, Sakshi, can you tell me what's a cell?
I've heard of it, but I don't know the meaning.
Okay. Janvi, can you tell me what is a cell?
I think a cell is like very small part, um,
Yes.
sort of
Okay. Very close. Okay.
organism
See,
which is
very
alive.
good. Okay. Yeah. So a cell is the smallest part of an organism. Okay, like we are made up of millions of cells. Okay, all the organisms are made up of many number of cells. For example, if you take a house, a brick is like the cell. For example, in the house, a brick is like the cell in our body. For example, our body is like a house. Okay, our body is like the house. Then how a brick is in the house, in the same way a cell is in our body. Okay, many bricks make up the house. So in the same way, many cells make up the body of an organism. Okay, so the bacteria is only having one cell. It's only having one cell. So it's called unicellular. Uni means one, cellular comes from the word cell. So it has only one cell. And it has a very simple cell. Okay, so that's called a prokaryotic. Uh, they are called prokaryotic organisms because they have a very simple body structure. Okay, they have very simple body structure, so they are called prokaryotic organisms. And bacteria, how do they get their food? Some of them prepare their own food, and some of them don't prepare their own food. The ones which prepare their own food are called autotrophic. The ones which don't prepare their own food are called heterotrophic. Now, tell me an organism which prepares its own food. And which organism prepares its own food? Living organism? No? Do plants prepare their own food? Yes. So plants do prepare their own food. Okay. So in the same way, yes, Janvi, you want to say something?
No, I just wanted to say that I think the organisms which prepare food for themselves are called autotrophs, right?
Yes, they're called autotrophs. Okay, so plants prepare their own food, right? So in the same way, some bacteria also can prepare their own food. But plants have a pigment. What is the name of the pigment? Can you all tell me? What is the name of the pigment due to which? Yes, Janvi?
Chlorophyll.
Chlorophyll. Very good. But bacteria does not have chlorophyll. But they prepare their own food by using some chemicals. Okay? Like um, plants prepare their own food by a process called photosynthesis. In the presence of light, they prepare their own food. But bacteria does not have chlorophyll, so they cannot prepare their own food by the process of photosynthesis. They need another methodology to prepare their own food, that is called chemosynthesis. Chemo refers to chemicals, and they break down the chemicals to prepare their own food. Okay, so some bacteria prepare their own food by chemosynthesis, and some bacteria cannot prepare their own food. So there are two types of bacteria. Some can prepare and some cannot prepare their food. Okay. Now there are four basic shapes of bacteria. You can see the picture, that is rod-shaped bacteria, which is called bacillus. Okay. Some, yeah, this is the rod-shaped bacteria, which is called bacillus. Some bacteria are like in the shape of a comma. So that is called vibrio. Some bacteria are spherical in shape, okay? Rounded structures they have. They're called cocci. And some bacteria, they are in the form of a spring. You all have seen a spring, right? A telephone wire coiled. So that is called a spirillum. So there are four basic shapes of bacteria. Okay. Now bacteria can be useful to us. They can also be harmful to us. Some bacteria are useful. For example, we can say the lactobacillus. It's a bacteria which is useful to us. It is a bacteria which is converting the milk into curd. So it is useful to us. Okay. There is another bacteria called the rhizobium bacteria. What it does, it changes the atmospheric nitrogen into usable forms of nitrogen so that the plants can take them from the soil. Okay, so that the plants can take them from the soil, and from the plants animals will get them, from the plants even humans can get them. So we cannot take in nitrogen directly from the air, we need to take it from the plants or the plant products. Okay, so we can say that rhizobium bacteria helps in nitrogen fixation. It fixes the nitrogen from the atmosphere into the soil. Okay, so these are the examples of two bacteria that are useful to us. There are many bacteria that are also harmful for us. They cause various types of diseases. Like what are the diseases they cause? Like typhoid, cholera. Okay, these are the diseases that they cause: typhoid, cholera, tuberculosis. These are the diseases that are caused by bacteria. Now we'll move on to the next microorganism, that is algae. Okay, algae are microorganisms that look like plants, but they are not actually plants. They look like plants. They don't have root, stem, leaves. They don't have any sort of thing. Okay, but they are green in color, so they look like plants. And why are they green in color? What is the pigment that they're having? What is the pigment that makes them green in color, like the plants? Yes, Janvi?
Do they have chlorophyll?
Yes, they have chlorophyll.
Okay.
Okay. So, as the plants are green in color because of chlorophyll, in the same way, algae also have chlorophyll. So, they are also green in color. Okay. Since they have chlorophyll, they can also prepare their own food. So, they are autotrophic organisms. Okay. Algae are in no way harmful for us. They are always useful to us. They are found as a thin layer on top of the pond, lakes. Okay, you all must have seen. They are called the pond scum or something. Okay, now they are green in color, so that is useful. They help in increasing the oxygen content of the pond so that the organisms get a lot of oxygen. Okay, pond scums are useful to them. And we also in some countries, algae is taken as a seaweed. um, I mean, they are eaten, they are edible. Okay, some algae, they are edible. Some seaweeds which are algae, they are edible. Okay, they can be eaten. Some algae, that is called the red algae, they are used in ice creams, they are used to prepare jellies. So in food industries, algae is used. Okay, in labs they have a jelly-like medium called agar-agar that is also prepared by algae. So algae is useful to us; they are not at all harmful for us. Okay. Now we move on to the next microorganism, that is fungi. Okay, fungi, they don't have chlorophyll like the algae, so they cannot prepare their own food. So how are they going to get their food? They are going to get their food either from a dead organism or from a living organism. If they get their food from a dead organism, okay, like a dead log, some fungi grows on dead logs. So if they're getting their food from a dead organism, they are called saprophytic. What are they called? Saprophytic. Yeah. And if they live on any living organism, means if they get their food from any living organism, they are called parasitic. Okay, so there are two types of, they get their food either from dead organisms or from living organisms. If they get their, yes Janvi, you have some doubt?
How do they take food from dead organisms?
See, for example, there's a dead log. Okay, in the dead log there may be many nutrients which was there in the tree. Okay, in the dead log there may be many nutrients. For example, if an animal dies, okay, now inside that animal there are a lot of nutrients. When there is decomposition of those animals, the fungi also grows there. So though the nutrients from the dead animal or the nutrients from the dead plants, it takes those nutrients and it lives. Okay, so that is how they get their food. If a plant dies or if an animal dies, you have seen decomposition, or you know that decomposition takes place, right? It breaks down, it mixes with the soil. So from that animal or from that plant, there are nutrients are there inside every living organism. So these fungi, they live on them. Okay, and they get their food from them. So if they get it from a dead organism, that is saprophytic. Sometimes they live on a living organism, maybe on a tree. You all must have seen, I'm not giving an example of a fungus, but you all must have seen some climbers on a tree. Some climbers, uh, you know, twinning around a tree. So they don't prepare their own food. Okay, they get their food from the big tree. You'll have seen small plants coming from the big tree. Okay, so they get their food from there. So that is like a parasite. We can give some examples of these worms live inside the body of organisms, some intestinal worms. So what do? Organism eats food and they get the food from the body of the organism. So they are parasites because they live inside a living body. Okay, so in the same way, fungi also gets its food either from a dead organism or from a living organism. If they get it from a dead organism, they call it saprophytic. If they get it from a living organism, they call it parasitic.
So mushrooms are saprophytic because
Yeah,
I while
mushrooms are
while
saprophytic.
I was walking, I found mushrooms growing out of a dead tree.
Yeah, so mushroom is saprophytic. Okay, and what are the examples of fungi? We can say bread mold. Okay, on a piece of bread, when the bread gets stale, when the bread gets rotten, we see some grayish or blackish or greenish patches on the bread. That's a mold. That's a bread mold. Okay, so bread is also a dead, means it is, when it gets stale, it is considered as a dead organism or a dead, you know, it's not living, right, the bread. So that is, a bread mold grows from it. Okay, Yeast is another example of a fungus. Now, what does the yeast? It also has many roles. Okay, it is used to make bakery products. Okay, where we see the bakery products, they rise. The dough rises. The bakery products are nice and spongy, fluffy. Okay, we get very spongy bakery products, cakes, pastries, buns. Okay, so that is all because of the yeast that we add to it, and yeast is a fungus. Mushrooms, as Janvi said, mushrooms are also fungus, but they are very big. We can see them with our naked eye. We don't need a microscope to see them. Okay, but they are also coming under the category of fungus. All right, and mushrooms are edible. All of you all know that. All right. Now we go to the next microorganism, that is protozoa. Protozoa is also having a single cell. They also have only one cell. We can see them under a microscope only. They are found mostly in the water bodies. They are aquatic. They are found in the water bodies, in ponds, lakes, ditches. Okay, we find them. Yes, Janvi?
Mushrooms are not micro, like they're not small. So why are they called micro? Why do they come in the category of microorganisms?
Okay, they're coming under microorganisms because, see, when the one who made classified everything, he categorized all the organisms. Okay, he found that mushrooms are having all the properties of the fungus except that it is microscopic. It is non-green. It is not green in color, so it cannot be called a plant. A mushroom grows like a plant, but it is not green in color. It does not have the chlorophyll. Okay, so we cannot consider it as a plant. The main feature of a plant is that it has chlorophyll pigment. So mushroom could not be placed in algae. It could not be placed in a higher category called plants also. Okay, but it has all the other things like it has some structures which are like the roots called the rhizoids. Okay, it has some thread like structures which are growing, that is called the hypha. So all these things were similar to fungi, so they placed it in fungus. Okay, only it is not microscopic, but other things are similar to the category of fungus, so they placed it in this category. Okay, they having every other thing like fungus, so they placed it in this category. Okay, and there might be some exceptions in every case. There might be some exceptions in every case, which maybe one character may not be similar. Okay, but still they are placed in that category because all the other characters are similar to that category. Okay, so that is why fungi means the mushroom is macroscopic. It means it can be seen with the naked eye, but still it is placed in fungus because all the other characters match with the characters of fungi. All right. Now we were discussing about protozoa. Protozoa are aquatic organisms. They are found only in the water bodies, in the ponds, in the lakes, ditches. We find these organisms. Okay. Protozoa are used in the treatment of sewage. What is sewage? Sewage. Anyone knows? Yes, Janvi. Tell me.
Um, Somewhere where there's wastewater.
Yes, wastewater. Okay, the wastewater that is generated from our households, the wastewater that is generated from a household, from the offices, from everywhere, from the industries, all this is called sewage. So sewage is being treated. Sewage is being treated by the help of protozoa. Protozoa helps in treatment of the sewage. Okay, it helps in treatment of the sewage. It means it breaks down these the waste materials in the sewage so as to make the water pure. Okay, in order to remove all the impurities from the water. Then viruses. Okay, viruses are the next group of microorganisms, and viruses are, we can call them the villains of the group. Okay, they are the most deadly of all the microorganisms. They are the most dangerous, they are the most nasty organisms in the group. Okay, because they can only be harmful for us. They are not at all useful to anybody. They are only harmful. And why are they so dangerous? Because they cannot be considered living or dead. They are coming in the borderline of living and dead. Okay, they're coming in the borderline of living and dead. When they enter into a living organism, they pretend as as if they are living, means they are living. But once they move out of the living organism, they seem to be dead. They show no life. So it is very difficult to say whether they are living or dead. It is coming in the borderline of living and dead, or living and non-living. Okay, and they cause types of diseases various types of diseases. You can say common cold, you can say fever, you can say measles, chickenpox, AIDS. So many. You all have heard about swine flu before, right? Swine flu was a disease.
I've heard about it.
Okay. So swine flu was caused by a virus called H1N1 virus. We knew that swine flu came from the pigs, but pigs had the virus inside. Because of the virus, swine flu occurred. Okay. And you all have heard about Ebola virus before any time? It was it killed so many people in Africa. Africa was completely, so many people had died because of the Ebola virus. Okay, what had happened means the Ebola virus had caused so many blisters on their body, and that led to death. Okay, the Ebola virus was very, very dangerous. In this AIDS virus, AIDS is also called a deadly disease. Okay, it leads to death. So many viruses are there which are very harmful for us. Okay, next slide. Okay, so viruses can be useful to us in so many ways. You all can see, that viruses are used in making curd. Tell me which of the five types is used in making curd, whether it's a bacteria or a fungus or a virus. Yes, Janvi?
It's a bacteria, lactobacillus.
It's a bacteria. Lactobacillus. Okay. So the lactobacillus bacteria, it changes milk into curd. So it's useful to us. Okay. Bakery products are made by one of the organisms. Tell me, what is the name of the organism that is used to make the bakery products, that is used to rise the dough? Yes, Janvi?
Yeast.
Yeast. Okay. So yeast helps in rising the dough. Okay. It helps in making these bakery products nice and spongy, soft and fluffy. Okay. Alcohols are also prepared because of a certain organism. What is that organism? Alcohols are also prepared by an organism. That is also yeast. Yeast also helps in making alcohol. Okay. And the dairy products are also made by the bacteria. Okay. So all these things, you all can see, the dairy products, the curd, bakery products and alcoholic drinks, they are all prepared by a process. What is that process called? Fermentation. They are all prepared by a process called fermentation. For now, you just remember fermentation. In the later slides, we will discuss what is fermentation. Okay. So dairy products, curd, bakery products, and alcohols are prepared by a process called fermentation. Okay. Now, microorganisms are also used in the medical industry. They're used in making vaccines. Okay. Vaccines and medicines are different. We eat medicines when we are sick. When we get the disease, after that we eat the medicines. But we take in vaccines before the disease so that we don't get the disease. Okay. So how does a vaccine work? I'll just tell you all. A vaccine, when it when you put a dead or a weak microorganism into the body in the form of an injection, or you eat it orally as a tablet. Okay, what will happen when something from outside, for example, our country is guarded by the Indian army. Okay, now suppose someone from outside enters a country. What will the army do? What will the army do if somebody, some other, uh, some person from outside enters a country, or a group of people from out of India enter the country? What will the army do? They are dangerous for the country. Yes. What will the army
Attack.
do? Yes, they will attack them. In the same way, when microorganisms enter our body feels, okay, this is something from outside. Microorganisms are not from inside our body. They have come from outside. Okay, so they prepare weapons for it to kill these microorganisms. They prepare some weapons against it. What are these weapons called? They are called antibodies. They're called antibodies. Okay, so when the real microorganism, this is just a demo. Okay, so antibodies have already been prepared inside the body. Now, when the real microorganisms causing the disease will try to enter, what will our body do? Our body will attack with those weapons which have already been formed in the body. Okay, so our body will not allow the real microorganisms to cause the disease in our body, because before causing the disease, they will attack them with the weapons called the antibodies. This is how a vaccine works. Okay, this is how a vaccine works. A vaccine is just a demo of the microorganism, and our body prepares the weapons against it. When the real microorganism enters, then the weapons are, they attack with the weapons. Our body attacks those microorganisms with the weapons that they have created. Those are called the antibodies. Okay, so in this way a vaccine works. Okay, so they help in preparation of vaccines. I also told you all that they help in they help in treatment of sewage. Sewage is a wastewater. So bacteria and fungus, they help in decomposition, and even protozoa helps in, they help in clearing the sewage. That means they help in removing the impurities from the sewage. Okay, many types of organic acids, we can say vinegar is an organic acid. We use it in our kitchens. Okay, it's also used on a large scale. So vinegar is an organic acid which is produced by the microorganisms. Citric acid, different types of organic acids are produced by the microorganisms. Microorganisms also help in decomposition. Decomposition, you all know, when anything dies, maybe a plant or an animal, then that is decomposed, that is mixed with the soil. So what helps in the mixing? The bacteria, fungus, they help in decomposing the organism, okay, with the soil. Okay, so these are some of the useful effects of microorganisms. Okay, next slide. Okay, now we talk about fermentation. We discussed in the previous slide that dairy products, curd, alcohols, bakery products, they are made by a process called fermentation. Now, what's this fermentation? It is the process by which the microorganisms break down something in the absence of oxygen. Okay, they are breaking down something, but this is very important that it is in the absence of oxygen. Fermentation occurs always in the absence of oxygen, when there is no oxygen. Okay, so breakdown of food or breakdown of any type of food in the absence of oxygen, that is called fermentation. Okay, now there are some examples of fermentation. Like we said that milk is converted into curd. What happens there? There in the milk there is a sugar called the lactose sugar. The lactose sugar is broken down to form lactic acid. So that is a process of fermentation. And it, okay, that is a process of fermentation. process of fermentation. Another example of fermentation is when yeast breaks down sugars to form alcohol. Okay, yeast also, we have read that yeast helps in preparation of alcohol. So what it does, it breaks down the sugars to form alcohol. What does sugars mean? It means the carbohydrates. It breaks down the carbohydrates to form alcohol. Okay, so this is fermentation. You can see in the picture, when sugar combines with yeast, the yeast breaks down the sugar to form alcohol and carbon dioxide. So it produces alcohol and carbon dioxide. And because of that, so many things are produced. See, alcohol is used in sanitizers, you all know alcohol is present in the sanitizers. Alcohol is also used in some medicines. Mostly in the homeopathic medicines, alcohol is used. Okay, alcohol is used in the labs for various experiments. Okay, we use it in the labs. Okay, apart from alcohol, carbon dioxide is also produced. You can see that in the soft drinks. When you open the soft drinks, what happens? It gushes out. Okay, because of the carbon dioxide which is present there. Okay, apart from alcohol and carbon dioxide, lactic acid is also produced. Now, lactic acid is present in curd. Lactic acid is present in other dairy products. Okay, so in this way, fermentation is useful to us in many ways. Okay, it is useful to us in many ways. Next slide. Now, how these microorganisms are harmful for us? The most, how is it harmful? It can cause various types of diseases. It can cause diseases in plants, in animals, and in humans also. You can see the plant diseases that are caused by microorganisms. Have you ever eaten sugarcane? And you must have seen something red sometimes in the sugarcane. Some portion of the sugarcane is red. Okay, now that is called red rot of sugarcane. That's a disease caused by a microorganism. That's the disease of the sugarcane. You must have seen spoiled potatoes like this. This is called late blight of potato. Okay, in the citrus fruits like the lemons, oranges, you must have seen these brown color spots. Okay, so that is called citrus canker. In the wheat, you can see the wheat, that strand of wheat has become black. Okay, so that is called the wheat rust. So many types of diseases are caused. Many types of diseases are caused in plants. You can see the pictures of some of the diseases caused in plants. Many types of diseases are also caused in animals. uh, in cattle, anthrax is caused. Anthrax is a disease for the of the animals. Anthrax, black water is also caused. It's a big sore in the cattle animals. Okay, a big sore is created. That is open like a wound. Tuberculosis is also caused in animals because of microorganisms. You can see some of the diseases in humans that is caused by microbes. The most common thing, the first one is common cold. Yes, Janvi, any doubt?
Tuberculosis can also happen to animals.
Uh, what, Janvi? I couldn't hear
No,
you.
you said,
Tuberculosis.
yeah, you said that, and some animals also
Yeah,
have tuberculosis.
yeah. Tuberculosis is also caused in animals. As it is caused in humans, it's also caused in animals. Okay. Yeah. So you can see the first picture. It's the picture of common cold. Common cold is caused by a virus. Okay. The next picture is that of measles. Measles is also caused by virus. The next picture you can see of ringworm. All the types of skin infections, the skin diseases, are caused by fungus. Remember all the time, most of the skin infections are caused by fungus. If ever you have got a skin infection, and you'll have visited a doctor, they might have said it's a fungal infection. Fungal infection means it's caused by a fungus. Okay. Then you can see malaria. Malaria is caused by a protozoa. What is the name of the protozoa? Plasmodium. You all might think that it is caused by mosquito, but mosquito is not causing the disease. Mosquito is carrying the plasmodium. When it bites us, it is transferring the plasmodium into our body. That's how we can get malaria. Okay? It is only the carrier of the disease, but it does not cause the disease. It only carries the disease. It is only carrying that protozoa inside it. When it bites us, it is transferring the protozoa into our blood, and that's how we can get malaria. Okay, so these are some of the diseases that are caused by microorganisms. There are multiple number of diseases that are caused by microorganisms. And remember one more thing, that the diseases that are caused by microorganisms are always communicable. What is the meaning of communicable? Means they can transfer from one person to the other. The diseases that are caused by microorganisms are communicable diseases. They can be transferred from one person to the other. Okay? All the diseases that are caused by microorganisms are transferable. They can be transferred. All right? Next slide. Okay. Apart from that, microorganisms also spoil the food. They also destroy the agricultural crops. Okay? So, in order to prevent the spoilage of food, we use certain techniques called food preservation techniques. Okay? See the first technique, that is called sun drying. That means we can dry some fruits in the sun. We can dry some food items in the sun, and they can remain for a longer time without getting spoiled by the microorganisms. Okay, they will remain there. They will remain as such for a longer time. Dry fruits. All of you, both of you must have eaten dry fruits, right? You all know what are dry fruits. Okay, dry fruits, they remain for a longer time. Okay, you all know that they don't get spoiled easily. They remain for quite a very long time because there is no water inside them. They are completely sun dried. Okay, different types of fruits can be sun dried. Then we go to the next one, that is boiling. When you boil milk, the milk remains for a longer period of time after boiling. And if we don't boil milk, the milk will get spoiled faster by the microorganisms. So boiling is also a food preservation technique. We also add various types of chemicals. Chemicals like sodium benzoate, benzoic acid. We add different types of chemicals also to the food. Like we add chemicals to the jam, jellies, squashes. Different types of preservatives are added. If there were no preservatives, they would have got spoiled very soon. But you know, squash, juice, and all, they remain for a very long time. Okay, if you keep it out of the fridge also, it will not get spoiled. It's not going to get spoiled because they're having preservatives. They're having some preservatives. Whenever you all see a juice bottle or a squash, the bottle of a squash bottle, you'll read the ingredients. You all will find some preservatives there. Okay, you all will find some preservatives. Preservatives are added. Cooling is a very common food preservation technique. We keep things in the fridge so that it does not get spoiled easily. Fruits, vegetables, milk, all is kept in the fridge so that it does not get spoiled easily. Okay, we provide a lower temperature to it. Pickles are also made. Raw mangoes may get spoiled, but when you make raw mango pickle, that remains for a very long time. It can remain for about 5 to 6 months also. Okay, it does not get spoiled. Salting is also a food preservation technique. You all must have seen salted fish. Salted fish, it is dry. Okay, no, what do they do? the fish is completely dried and salt is added. Salt is added. When you add salt to something, you will see the water from that organism, it comes out. If you add, you can try it with any vegetable. Okay, suppose you cut a piece of brinjal and you put salt on it. After some time, you will see water is coming out from there. Some water is coming out from the brinjal. Okay, it happens if you put salt. If you put salt, then water, the organism gets dehydrated. The water starts coming out. And when there is no water, microorganisms have less chances to survive. Okay, so salting is also a food preservation technique. We also add sugar, like the jams, jellies, they're loaded with sugar. Squashes, also sugar is added. Okay, so that also can keep the food for a longer period of time. Deep freezing is also done. We can keep food items in the deep freezer. Fruits, vegetables can be kept in the deep freezer. We can also add vinegar. Suppose you are going traveling, okay, and your mother has prepared some food and you want it to remain for a longer period of time. You can add 2 to 3 drops of vinegar to it. That will keep it as such for a longer period of time. Okay, so adding of vinegar can also preserve it for quite a long time. And then canning. When you must have seen that in the malls and all, uh, they sell products in airtight containers. Okay, so that is preserved, means that it is packed in airtight containers so that they don't get spoiled. Many things, dry fruits are packed, um, different types of packed food items, this is all by canning. Okay, they can be preserved by this method. And another important method is pasteurization is only done for milk. What they do, when the milk is taken out of the cow, okay, when the cow gives the milk, it is heated to a particular temperature, and after that it is suddenly cooled. So when it is heated, the microorganisms die, and when it is suddenly cooled also, the microorganisms which could survive in the cold, they are sorry, which could not, like, the microorganisms get killed in this process. Sudden heating and cooling. Okay, when you heat it and suddenly cool it, the microorganisms all die, and that's how we get the packet milk. Nowadays we get packet milk. No, that is pasteurized milk. That's pasteurized milk. Okay, we are getting the pasteurized milk, which is already pasteurized in the industries, in the milk industries, in the dairy farms. All right, next slide. Okay, now we'll carry out the experiments. So, see, the first experiment is to study the microorganisms. Okay, the first experiment is to study the microorganisms. For that, you will need, see, I have collected soil. Can you see it? Yes. So I have collected soil and I have put water to it. Okay. What happens? The soil settles down and the water remains on the top. Now, what we need to do is, I'll show you what to do. You all cannot perform this experiment, but you all can see how it is done. Okay. I cannot show it under the microscope also to you all, but I'll show you all how it is done. So what you all can do, you all can take a drop of this water and you all can add it to your slide. One drop. All right. Now this drop may have lot of microorganisms in it. Now what you do, can you see the drop of water here? I have added a drop. Okay. Now you can put, this is called a slide and this is called a cover slip. All right. This is called a cover slip. Now to the slide, you can put the cover slip. How will you put it? You can put it from one side like this. Can you all see it? You can put on your I mean, you can unmute yourselves so that I can hear you all, because I cannot see you all.
Yeah.
laptop.
Yeah.
Yeah,
Nicola ma'am, because of the white background, it's not visible. Can you put it on the black portion?
yeah. Is it okay?
Yeah, much better.
Yeah. So what happens? So this is a cover slip. Okay. Now what we do? We put it down like this slowly. Sorry. So I'll show it to you all once more.
Swapna ma'am, can you stop sharing because then it's coming really small. Yeah, thank you.
Yeah. So again, I'm showing you all. You all can put a drop. Yeah. And now what you all can do? You all can put this cover slip slowly on it. Why do we put it so slowly? So that air bubbles will not enter. And then what you all will do? You all will take a microscope, okay, and you all will place it on the stage. This is called the stage, okay. You all place it on the stage, and then you all can see. You all can see what are the organisms that are present. Okay, in this way you all have to do it, but I cannot show you all what we can see in the microscope. But this is the way that you all have to perform this experiment if in the lab. Okay, you all can see it under a microscope. All right, now the next experiment. You all can take pond water or you all can take muddy water and add water to it. uh, Sorry, you all can take either pond water or you all can take mud and add water to it, not muddy water. You all can take mud and add water to it, and that water you all can see, okay, under a microscope. Okay, the next slide. See, you all can see what are these? These are protozoa which are found. You all can see these type of organisms in the water. Okay, so many protozoa can be seen in the water. Okay, you all can try it with pond water. You all can also try it with moist soil, and you all can add water to it. Next slide. Yeah. Okay. So here we will see how dough is how the dough is fermented. For that, you have to take maida or atta. Okay. You have to take some sugar, and you have to take yeast. You have to take containers also. Maybe one container will do. One container you all need to take. You all need to take maida or atta. You all need to add 2 tablespoons of sugar to it. A little bit of atta you all can take. 2 tablespoons of sugar to it, and then you all can add yeast. So what you all will
Okay.
do, first of all, but you all
Uh,
need lukewarm water. You all need lukewarm water, okay? In the lukewarm water, you all add the sugar, little bit of lukewarm water, not much. Little bit, add sugar and add the yeast to it. You all can
ma'am,
show, you all can show us what you're
they
doing
can perform the activity with you right
after
now.
the class.
No, they can
Okay,
perform it
now,
right now, and then they can observe the results and send us pictures.
okay. See, I want to show you all how what will it look like. Okay, see, this is the dough without yeast. Okay, this is the dough without yeast, and this is the dough with yeast. So you all can see it has risen a bit. Okay, what you'll have to do is you all need lukewarm water. You all need warm water for that. You'll take a small container. I'll instruct you what to do, and then don't you'll have to take warm water. You'll have to add sugar and yeast to that water.
Ma'am, my warm water is gone cold. Can I go warm it up
You can
again?
quickly go and get warm water, Sakshi.
Okay.
And Janvi, please turn on your video.
Yeah, I just gone get the water.
Yeah. You need warm water. In the warm water, you add 2 tablespoons of sugar and yeast. Okay, if it is such a big spoon, then you can add 1 tablespoon. Okay?
It's a tablespoon. This is the
Okay.
measurement of a
All right.
tablespoon.
Okay. You have to add it to lukewarm water. Okay, Janvi? Yeah. And then you add yeast to it. Okay.
Please dissolve the sugar before adding yeast.
Yeah. Now you can add a little bit of yeast.
What do I have to do first?
Yeah. First, you take a little bit of lukewarm water. Okay. Add 2 tablespoons of sugar to it. Yes, actually, you can adjust your camera so that we can see what you're doing.
I am on the computer and I can't adjust it.
Okay, fine. So you can lift it up and show us what you're doing. Otherwise, because are you and have you added 2 tablespoons of sugar?
Yes.
Okay, then stir it and dissolve it completely. Okay. Then you can add yeast to it. Okay. You can add yeast and again stir it. After dissolving the sugar, then you add the yeast and stir it again.
How much yeast shall I add?
Um, one tablespoon.
Okay.
Have you taken little water, Sakshi? Or you have taken a lot of water? Little water?
I don't know how to describe it. I think a lot.
Yeah,
Show. Just you can you lift it up and show us?
I think it's like this is
Yes, Sakshi.
my thing, and I felt it till here.
Okay.
yeah. I added the yeast. There was no internet, so
Okay. You added yeast. Now you stir it. Janvi,
yes.
stir it.
Okay.
Okay. Are there bubbles forming there?
Yes.
Okay.
Okay. I don't.
Now what you can do, you can take atta or maida, whatever you have. Okay. All right. Yeah. And you can add little bit of water, and how your mother mixes and makes a dough.
One.
You can add.
We will, Nicola ma'am, 1 second.
Yeah.
We will give them 5 minutes time for the yeast to get
Yeah.
activated, and we will explain
Okay.
them what is the process, and then they can add
Yeah.
atta to the water. Let the yeast get activated.
Okay.
Yeah. Okay. So
So after that, when the yeast gets activated, then you all can make a dough. You all know how to make a dough. With that water, you all have to make a dough. No, you all must have seen your mom making a dough. You all have to just put water and mix it up. There's you all need to take a little bit of atta. Don't take lots of that. Okay, a little bit of atta or maida you all take, and you all have to knead the dough. You'll have to mix it up with that water. Okay? Okay. Yes, actually, you can do it. But y'all can, after 5 minutes, y'all will do it. Just see the time, and after 5 minutes, y'all can start the process.
yeah. So in that till that time, we'll just Nicola,
You
I'm
can,
just sharing my screen so you all can see
yeah,
what
I
the
can move on with the
experiment
next
is all
experiment.
about. Yeah.
Yeah, yeah. Okay. Okay. So this shows how the dough rises up after you keep it for about 2 to 3 hours. All right. Next slide.
We just made a pizza dough last week with the same process.
Okay. Very good. So, so did you see that the dough had risen up, Janvi?
It was
Yeah.
just big. It was bigger than the container.
Okay. Okay. Next slide. Our pictures have frozen. Okay, something is wrong with
I can
mouse.
see all of you all moving and laughing.
We can't see the screen.
You can't see the screen.
No.
You can't see my window.
We can see it, but it's frozen. It's like stuck.
Now,
Yeah. No, we cannot see any slide.
My yeah.
1 second. I'm just starting it again. Better? Is it visible?
Yeah, it's okay.
Yeah. Okay. So, as a result of fermentation, carbon dioxide is also produced. So, what you'll have to do, you all can take 3 containers, okay, whose mouth is narrow. Do you have any such containers? If not three, two will do.
Hmm. They can use
Which has a
those
narrow mouth, maybe a bottle you
Pepsi
can take.
bottles. Pepsi bottles.
You all can take bottles, okay. Small bottles will work for this experiment. Do you have them right now?
Will this work?
Yeah, it will, but yeah, it will. But that's a little bit big. If you then you have to add more of stuff. Means more of maida, sugar, and yeast
We only have
in order
one.
to see the carbon dioxide. Do you have small bottles, this size ones?
I only have like
Smaller one?
Is this okay?
Yeah, that will do. That will do. Sakshi, that's going to do. Even if you all have one bottle also, that will serve the purpose.
okay. I'll just go and
If
get.
you have one bottle, that will also work. Okay, you'll just have to see that carbon dioxide is produced in the experiment. So I'll tell you what to do. If Janvi, do you have a smaller one?
She's just gone to get one.
Okay.
In the meantime,
Yeah. See
girls,
what?
girls, you all think about why did you mix the yeast in warm sugary water? And you have to answer to Nicola ma'am. Come on, Sakshi.
Yeah.
Okay, Sakshi, tell me what happens in the process of fermentation. What is happening there in the process of fermentation?
I'm sorry, I'm not able to hear you.
Uh, I asked what is happening in fermentation. What takes place? What happens in fermentation?
Can I rejoin? I'm not able to hear anything.
Yeah. Okay.
Yeah, okay. So Janvi, uh, Janvi, while you were away,
Have you got?
a question was posed. Now, why do you think we add warm water and sugar to the yeast? Yes, you can
Yes,
answer.
Nihal, say.
Yeah. I think it could be so. It's like that temperature in which the yeast can grow, and sugar is sort of the food which it can eat to grow on.
Yes, and sugar is broken down, okay, to form alcohol and carbon dioxide. That is by the process of fermentation. Okay, good. Let Sakshi come. I'll just explain this experiment.
I found a
Uh,
bottle.
okay, you found the bottle. See, what you have to do is, okay, you're having a conical
Yeah.
flask. Very good.
Sorry.
So,
Yeah.
yeah, so you can you can add maida or atta. Okay,
How much maida shall I add?
uh, you can add a very little, maybe. Okay, y'all can add 2 tablespoons. See what y'all will do. Wait, one second. Before that, y'all can first y'all can take a little bit of lukewarm water, add sugar, and add yeast as y'all did in the previously. Okay, for the previous experiment. So y'all can take lukewarm water, add sugar, and add yeast to it, and then y'all can add the atta or the maida and stir it. And then what you'll have to do, you'll have to put a balloon to it, and if it remains like that, it is well and good. Otherwise, you can secure it with a rubber band.
Do I put the yeast and water in the bottle first and then put the maida?
Yeah. First you'll take lukewarm water,
I'll have to
add sugar
warm
to it,
water.
add sugar and stir it. Then you'll add the yeast to it, allow it to activate, and then you'll add the maida, and then put balloons. Okay. See what I have done. I have taken 3 test tubes. In the first test tube, okay, I have added maida, I've added water, sugar, but no yeast. Here there is no yeast. So y'all can see the balloon. It is not inflated at all. Okay. In the next test tube, what I have done, I have taken lukewarm water, I've taken sugar, I have taken yeast, I have taken little bit sugar here, not much. So y'all can see the inflation is very less. Okay, little bit. But here I have taken more sugar. In this case, I have taken more sugar. So you all can see it has inflated more. Yes, this is because of the carbon dioxide that fills the balloon. Where there is no yeast, no carbon dioxide will be formed. Where there is little sugar and yeast, less carbon dioxide will be produced, and where there is more sugar, then more carbon dioxide will be produced in that case. So this has been inflated more. You can see the difference, right? This is less sugar and this is more sugar. Okay, so that's what you'll have to do. You'll have to see, this is to show that carbon dioxide is produced during the process of fermentation.
I'll just go and get my loop.
Yeah. So now y'all can knead your dough. Okay. We will stop today at this activity. Okay. You perform your activity and observe the result and share pictures with us. And
Okay, you can knead your dough with
okay.
that lukewarm water and whatever you have prepared it. already. So with that, you all can knead the dough. And this experiment also you all can do the production of carbon dioxide. Okay, by fermentation. Yeah.
Okay.
Okay. In the next
Yeah.
class, we'll perform the other experiments. All
Yeah.
right.
So I wanted to ask Janvi and Sakshi, uh, 4 to 5 in the evening, which day is suitable for y'all?
For
All
me, any day after. For me, any day after, except Thursday.
Okay. And for you, Janvi?
any day except Saturday.
Any day except Saturday. Okay. So we'll finalize the day and we'll put a message on the group by tonight. Okay.
Okay.
And it'll
So
be 4 to 5
you
in
all
the
are
evening.
comfortable with Wednesday?
Wednesday is there might be a problem because I have a class exactly at 5.
Yeah. Even for me also Wednesday will be little bit of a trouble. So we can
If
do it
it's
on
on Friday,
Friday.
then
Fridays.
Friday is it good for both of y'all?
I have a class at 4 o'clock on Friday. I am free all days. It's not on Saturday. I think it's on yeah. I'm free on all days except Friday and Wednesday.
Friday, what time do you have a class?
4 to 5.
So is 5 to 6 good for you?
The problem is that class might get extended 10-15 minutes. So
5:30 to 6:30. Does it suit? Or it'll be too much of a back-to-back thing?
yeah.
Okay. Then, okay, we'll just finalize. So, except for Fridays, you are okay with any day, right, Janvi?
Friday and Wednesday.
Friday and Wednesday. And Sakshi, you are okay with any day, 4 to 5.
Any day except Thursday.
Any day except Thursday. Okay. So, we'll figure it out and we'll send a message to both of you on the group. Okay. You all carry out these two activities. Do you all have a microscope by any chance?
Oh, I think I have one.
Okay. Okay, for Sakshi, whenever we meet in the lab, we'll do the activity. And for Janvi, you can carry out the first activity. You can collect the muddy water from the garden and you can try to observe the microorganisms in that water. And other two experiments you have to perform and you have to send us the results. Yes. So for the dough, before and after, like, when you need it, what is the how big is it, and after 2 hours or 3 hours, how much it has become. So that difference you have to send us. Okay. All right.
Okay.
Okay. Thank you. Thank you, girls.
Okay.
Thank you, Nicola, ma'am.
Okay. Bye.
Bye
We'll meet
bye.
up next time.